Okay, so we're going to start. To open this forum, we have our wonderful science writer, Michael Ray, one of the originals of the foundation. He will give an introduction to our science and talk about the power of the SENS damage repair heuristic to deliver iterable extension of healthy lifespan. You can read his bio on our events page, and let's welcome Michael to our stage.
Um, so yeah, I'm doing an introduction to the session today, and my basic plan here is to talk to you about the overall SENS plan and then introduce the speakers. So everyone here, of course, is already probably recognizes that aging is a horrendous medical problem that can and must be solved. I think a lot of folks, even though they are supportive of that broad goal, uh, tend to lose track of what the specific strategy that SENS is, right? SENS Research Foundation is called SENS Research Foundation because we're doing research on SENS, which is not just the general idea that aging is bad and we should solve it, but a specific strategy to go about systematically eliminating the aging process from the human body. Um, so let me go through that. So this is basically the schema by which people age, and basically everyone who is in the field agrees with this so far. So we have a series of metabolic processes that keep us alive. This is everything from the part of metabolism that is usually called metabolism. So what we mean by metabolic rate in terms of processing energy from food into energy that does cellular work. But it's also things like the circulation of fuels. It's also deciding on when cells divide, deciding on how neurons will interact with each other, even activating something like apoptosis. Everything that the body is regulated to do is part of metabolism. But all of those are physical processes. And just like all other physical processes, they have unintended consequences that result from them. This is damage. So these are biochemical processes. These are side effects of the metabolic processes that keep us alive. And those cause damage to the cells and biomolecules of our bodies. And because the cells and biomolecules of our bodies are exactly the machines that allow our body to do everything that it has to do, damage to those units renders them dysfunctional and reduces our capacity to do the things that those tissues and organs are intended to do. And once that reaches a critical threshold where you don't have the reserve of those functional units anymore, then you have pathology. And this is what we usually think of as diseases of aging. What they really are is the manifestation of aging, that is, the accumulation of cellular molecular damage in a particular tissue, causing the loss of function of that tissue. That's what age-related disease is. Um, so the question is, like, everyone recognizes, at least when you get to the stage of pathology, that that is a problem. What are you going to do about it? And the conventional way that medicine has dealt with this in the past is what Dr. DeGray, our scientific founder, used to call geriatrics, what I'm going to call standard of care medicine. So this is, you have a disease, we recognize you have a disease. What can we do to prevent that disease from getting worse and killing you? So this could be anything from like emergency room medicine, like get you to the cath lab and stop this heart attack from killing you acutely, to you've already had a heart attack, let's give you aggressive use of statins and maybe a blood thinner and whatever drugs we can do to prevent the pathology in your body from leading to your death or further disability. Um, that it's obvious just on its face that it's not the best solution to this problem, but also uh, it's limited by the very nature of the aging process we've been talking about, because even as you are trying to avoid the worst consequences of that age-related disease, more damage is continuing to accumulate because aging is continuing to happen. And so, you're really fighting a losing battle. So there's another approach that has been around for quite a while, and that is both what is the best of today's preventative medicine. So, getting people on statins before they've had a heart attack and getting you to do exercise and eat well, and all the kinds of things that you would want to do to prevent the excesses of metabolism from, being more severe than they have to, but also most of what we would today call geroscience. And so the idea of looking at what are the cellular processes, what are the biochemical pathways that lead to aging damage, can we modulate those pathways in a way that will cause the accumulation of damage to go more slowly and thereby forestall the accumulation of pathology? Um, I think we'd all agree that is a better solution than waiting until you already have had a heart attack to intervene. But there are limits to it because, metabolism by its nature is there to keep you alive. And so when you are interfering with those processes, you may be able to do so in a way that is less damaging. But first off, you can't eliminate the production of damage, because to do that would be to eliminate the processes that keep us alive, and also because simply reducing the amount of damage that you are laying down going forward does nothing to remove the damage that is already accumulated. And so if you take a person who is already 50 or 60 years old, even if they are nominally healthy, they are still not as good in terms of their cellular molecular structure as they were in their 20s. And certainly if you already are at a stage where you have, calcification in your arteries, or where you already have a significant amount of beta amyloid in your brain, it's really not adequate to just say, well, we'll slow down the rate at which we lay down more of that, right? So Dr. DeGray's insight, and this is the birth of the SENS process, was to say there is another mode of attack that we have been ignoring this whole time, which is to directly attack the damage itself. Um, and the advantages here are on both sides of this equation. So we are leaving metabolism to operate normally, and we are not interfering with the processes that keep us alive with all the attendant side effects that doing so has. But we are also not waiting until you already have pathology, you already have your health compromised to deal with it. We are attacking the damage directly. And because damage is by definition, abnormalities in the carefully designed structures of the cells and biomolecules of your body, it's by definition pathological, and virtually by definition, removing that can be nothing but good. And so it is a much more powerful intervention because it can actually remove damage after it's already accumulated. And it's a much more benign approach because we are not interfering with the processes that are there to keep us alive and functioning day to day. We are just restoring you to the cellular and molecular structure and health that you had in youth. And SENS has a categorization system for this, where Dr. DeGray just went systematically through all the different kinds of cellular and molecular damage that accumulate in the body by category. Um, in recent years, an alternative schema called the hallmarks of aging has become popular. It is good that they are taking that kind of divide and conquer approach, but it's a bit of a dog's breakfast in terms of they are using a mixture of, actual aging damage with things that are reaction to damage, the things that are processes that respond to damage, things that are downstream, things that are upstream. It really doesn't make an effective way to attack the problem. Whereas SENS goes to the root of the problem, looking at the damage and saying, how are we going to remove, repair, replace, or render harmless all of that damage? And then from that, the adaptations, the metabolic changes, all of that stuff will be reset because form follows function. Once you have restored the form, the structure, uh, the function follows afterwards. And so if we look schematically about how we would see this all working, this is the way that the geroscience approach, or the preventative medicine approach, works. So you are going about your life, and we now change the rate at which damage accumulates. That gives us a extension of life before we reach the threshold of pathology and frailty, and then eventually death. Um, that is a better approach than the default in red. It doubles the remaining health span, but it only raises the total amount by about 20%. What if instead we say, instead of slowing down the rate of damage accumulation by 50%, we remove half the damage? Well, here at the same point, we are actually doubling total health span instead of remaining health span, and we are boosting the remaining health span by fivefold. And that is simply because we are not just dealing with the accumulation of damage going forward, we are actually removing damage that has already occurred. So it is a very powerful approach just right off the bat. And then, of course, like any kind of science or technology, you can improve on it. And so if we come back after one cycle of this and we say we have a second generation of technology where we are able to further reduce by 50% of the 50% that we are already able to remove, then we get not as dramatic an initial effect, but because it adds on to the effect that we have already achieved, we get a very large reduction in the total effect on lifespan and health span going forward. Whereas if we attempt to do that with the messing with metabolism approach, with the geroscience approach, you can see that, even if you could, in principle, slow down by a further 50% the rate of damage accumulation, you don't really get much marginal benefit out of that. And it's just not plausible that you're going to be able to do that because, again, metabolism, the processes that you are modulating are the same processes that keep us alive, right? So if I if you have a circulating blood glucose of 130 milligrams per deciliter, and I cut that down by 30 milligrams per deciliter, I have done you a world of good because without that excessive blood glucose, you have less of the abnormal intracellular processes, you have less of the accumulation extracellularly of dislocation end products, you are at much less risk of things like diabetic retinopathy, terrible damage to the eyes, you are less damage of losing sensation in your feet or your fingertips, or of having an amputation of, these days, typically a toe or a finger. Back in the day, that could have been an entire limb. Um, but if I try then to say, okay, that was great, let's cut them down by another 30 milligrams per deciliter of glucose. Now you're down at 70 milligrams of deciliter of glucose. Now you're in constant danger of a hypoglycemic crisis. And that is just not a good solution to this problem. Um, so that is why the SENS damage repair approach is the way that, first off, we're going to get much better gains in the short term, but in the long term, we have this very powerful iterable approach, and that's how we're going to get to longevity escape velocity, to actually keeping aging indefinitely postponed. So a couple of slides earlier, I showed you the range of damage that SENS is approaching. And today we're here to talk to both our internal scientists at SENS Research Foundation and also several scientists that I am really delighted that we are funding externally to do exciting research um, in the SENS approach. And so I'm just going to give you a brief introduction to some of those speakers today. Um, So the first speaker I'm going to introduce is working in the apoptoSENS area. This is probably the one area of SENS that has made the most dramatic and obvious progress. This is, broadly speaking, in the area of senolytics. So apoptoSENS is not just senescent cells, but it's a range of abnormal cells that accumulate in the body over time and are dysfunctional. And senescent cells is like the best understood category of such cells. And so the solution for SENS is to destroy those cells, overcome their death resistance. Um, so at the SENS Research Foundation Research Center, Dr. Amit Sharma has been working on principally immune-based approaches to dealing with this problem. So as many of you may know, it's been known for some time that senescent cells aren't simply left to their own devices in the body. The body's immune system does have a system for what is called immune surveillance of these cells. So immune cells going around to destroy these cells before they accumulate. But of course, they do still accumulate with age. And so how can we either rejuvenate the immune surveillance of senescent cells if that is going down with age? Or how can we enhance or weaponize that approach? And that has been a lot of what Dr. Sharma has been working on for some years. Um, so today he's going to talk to us about several lines of work he's been doing in this space. One of which is working with CAR NK. So some of you will have heard of CAR T cells. These are T cells, a kind of immune cells that have been carefully weaponized to attack uh, mostly cancer cells by altering the receptors on the surface of the T cell so that they can go after kinds of cells that they can't naturally go after. Well, uh, CAR NK is using the same kind of technology, but applying it to a different kind of cell called NK cells. And the advantage here is that NK cells are already the kind of cell that our body uses to attack senescent cells already. And so when you develop CAR NK cells, you are both introducing new receptors that can go after senescent cells in new ways, but still retaining the existing inbuilt capacity of NK cells to destroy senescent cells. And so he'll be talking a bit about that. Um, he will also be talking about ways that he has discovered in the process of looking for new receptors that he could use to weaponize NK cells using this CAR NK approach. Um, he also has discovered peptides that have inherent senolytic activity, and so how he could use that and also a completely new sort of classic small molecule senolytic approach using a new vulnerability in senescent cells that he discovered in the process of doing unrelated work. Dr. Sharma, I would have to say, is very, very creative and observant when he makes a discovery. He sees better than a lot of scientists can how this can be turned into a therapeutic approach. And so just like a happenstance discovery in the process of his research, he drilled down into it and discovered a way that is looking very promising using small molecules to destroy senescent cells on the basis of that discovery. Um, and also, it's interesting because when you look at different studies of how the immune system surveys senescent cells, in different contexts, different cells seem to mediate most of the work. And so there's one set of cells that has been shown in models of tumors after senescence has been induced by chemotherapy, in a model of liver disease where you get hepatocytes that have gone precancerous. I can hear someone who is talking to me. Yes. Huh. I'm going to continue with my talk, even though someone is apparently trying to talk to me. Um, uh, so, Dr. Sharma, this paper back in 2012 predicted that there would be a kind of immune cell that would be responsible for cells that accumulate as a result of aging, as opposed to these other processes. And I think Dr. Sharma may actually have discovered it. I'm going to briefly pause because I think someone is trying to speak with me now. Huh. Pardon me? I'm welcome to continue. Okay. Sorry. Bit of confusion there. Um, okay. So the next area that we're going to be highlighting today is mitosens. So this is the problem that mitochondria, although they uh, cause a bunch of different functional changes with age, most of those are secondary to other kinds of problems elsewhere in the body. The one that is most clearly directly happening in the mitochondria themselves is the accumulation of mutations, and in particular, these large deletion mutations. Um, I'm delighted to say that Dr. Bhumanathan, along with colleagues here at SENS Research Foundation, after a long period of stagnation in the field, has been making really excellent progress in the last few years. Uh, Back here, this was several years ago, both showing for the first time that allotopic expression not only could restore the function of the mitochondria that had accumulated mutations in this mitochondrial genes ATP8 and ATP6, but really robustly showing that allotopic expression was actually doing that by restoring oxidative phosphorylation, which previous reports on allotopic expression have never clearly shown. This was the clearest and most decisive thing, and it really eliminated the skepticism in terms of is allotopic expression really working. She's going to talk a bit about her subsequent progress there, in particular on two genes, ND4 and COX2. Um, in addition to that, uh, up until now, all of this progress has been happening just in cells, in isolation. And of course, we want to work it eventually in living, breathing humans. Well, we have gotten the next best thing. Dr. Bhuvanathan has actually achieved this in living mice. So these are mice with a mild mutation in ATP8 through a combination of a technology that we developed thanks to Dr. DeGray's foresight and her allotopic expression work. She can now put the allotopic version of ATP8 into the cells of mice and show that it restores their mitochondrial function as we would expect from allotopic expression. She also has another, she's just going to touch on briefly. Um, Mitochondrial large deletion mutations occur because of a change in the way that the cell carries out quality control. So a lot of you will have heard of mitophagy, which is the process whereby cells recycle their mitochondria, eliminate abnormal mitochondria, and replace them with replication of the healthy mitochondria elsewhere in the cell. Well, it turns out that in the case of mitochondria bearing large deletions, this whole mitophagy machinery fails. And so they fail to see, it appears, the mutation bearing mitochondria, and they just accumulate and eventually completely take over the cell. And this obviously is a major problem. Um, in this new approach that Dr. Manathan will tell you about, she is working on a way to essentially eliminate that invisibility shield that these guys have and make them once again susceptible to mitochondrial turnover, mitophagy, and being replaced. And so that could not restore a cell that has been completely taken over by mutation-bearing mitochondria back to normal, because once it's 100% mutation-bearing mitochondria, there is no wild-type mitochondria to repopulate with, but rather, if you do this early on, you could forestall the date at which the cell becomes taken over and thereby extend the normal activity of that cell and bias time, basically. And Dr. Riba has been working for some time in iron metabolism aging. That actually began before he came to SENS Research Foundation to some degree, and he is now working on that further, looking at iron in a variety of contexts, and in particular in heme iron, the kind of iron that carries oxygen in our cells that we normally think of as being a safe storage form, but under some circumstances can become quite pathological. And this also builds on some work that was previously done at the SENS Research Foundation Research Center by Dr. Tesfahun Admatsu, who was our postdoctoral fellow, who discovered both the resistance of a whole class of senescent cells to most of the mainstream senolytic drugs, and also a new iron-based strategy to eliminate senescent cells. I am getting dangerously close to the end of my time, so I think I will stop, unfortunately. But there is also exciting work happening extramurally with Dr. Jean Hébert, repopulating the neocortex, um, and a project that I am very excited with that it took us uh, literally more time than would take a baby to get going with Dr. Annalise Barron at Stanford to develop new analogs of a protein in our body called LL37 that will have dual effects against Alzheimer's disease And finally, some work on the biology of lithium in the brain by Dr. Evan Snyder, who is just fabulous in terms of training many of the young rejuvenation researchers that we put out through SRF education program. I apologize to all three of you extramural researchers for not giving you your full due, but I'm very excited by everything that all three of you are doing and will look forward, as everyone should, to your presentations today.